The overall goal of this procedure is to identify the stages where viral gene expression is affected by chemical treatment. To do this, tissue culture cells are plated and incubated until confluent. The cells are then infected with either a triple reporter Vaccinia virus or a control promoterless virus. Next, treatment compounds are applied, and the cells are incubated for 18 hours to allow the completion of all stages of viral gene expression. The resulting fluorescence is then determined using a plate reader. Ultimately, results can be obtained that show the relative changes in expression caused by the experimental compounds.
The main advantage of this technique over infection with a virus that expresses a single fluorescent fusion protein is that this set of viruses provides more information about where in the viral life cycle a specific therapeutic drug is acting, providing insight into its mechanism of action.
Although this method can be used to identify inhibitors of viral infection, it can also be used to detect the stage of viral replication completed in non-permissive cell lines or in RNAi screens identifying cellular factors necessary for infection.
This protocol utilizes the triple virus, or TRPV, multi-stage reporter virus in which 3 spectrally distinct fluorophores are incorporated into the double-stranded genome of a single Vaccinia virus. Each of these fluorophores is controlled by a well-defined stage-specific promoter: the C11R promoter for early expression of Venus, the G8R intermediate promoter for expression of M. cherry,